Thanks for joining us for another episode of Biotech and Life Sciences CEO Stories. Today, our guest is another Atlanta luminary, Dr. Marvin Whiteley, co-founder of SynthBiome, a company using new technology as well as classic genetic techniques to address questions about microbial physiology, ecology, virulence, and evolution. Dr. Whiteley received his BS degree in zoology in 1995 from the University of Texas at Austin, and his Ph.D. in microbiology from the University of Iowa in 2001, followed by a postdoc fellowship at Stanford. After the postdoc, Dr. Whiteley moved back to UT Austin, where he was the professor of molecular biosciences and director of the LaMontagne Center for Infectious Disease. In 2017, he accepted the Bennie H and Nelson D. Abell Chair and the Georgia Research Alliance Eminent Scholar designation in Molecular and Cellular Biology at Georgia Tech. He also serves as Associate Director of the Cystic Fibrosis Center at Emory Medical School. So Marvin, I've got to ask you, just to start us off here, how do you go from zoology to microbiology? How does that happen? What inspired you there?
Yeah, well, I mean, it's kind of an interesting story, Tim. And thanks for the intro. I should get you to introduce me to my kids sometime so that they actually think we're doing interesting things. But yeah, I was actually an engineer for my first 2 years, and as an undergrad, I was aerospace engineering, and I was really enjoying it. And I was actually in a Fortran programming class. Like, we used to use this language called Fortran, and a professor came in who obviously had probably just had a grant that was not funded, or a paper that didn't get in, and came in, and there were only 6 of us in the class. He said, you should all quit, because none of this is, nobody's going to build airplanes anymore. You're just going to be mechanical engineers. So just go be a mechanical engineer. And this really like affected me, right? I was one of the first to go to college, for my family, and I was like thinking about this. And so I actually went out and started looking at other majors, and zoology actually in Austin at the time really appealed to me, because what it did is it allowed you to take classes in a lot of sciences. So you could pick geology, and I took physics, and I took microbiology, and kind of let you get a degree, but trying to see what you like while sort of staying in the hard sciences. And so it really was, while it sounds like zoology, might be something that's really far from microbiology and infectious disease, in Austin at the time, it really wasn't. And so it was an easy transition after that.
Interesting. Okay, good. So before we talk about the research itself, I thought we might kind of frame its importance with a little discussion about antibiotics and their current limitations.
Yeah, sure. I mean, I think everyone sort of, is hearing about this, if you're whether you're in the hospital, or, if you go visit someone, antibiotic resistance in the hospitals and in the clinics is something that we're really worried about. Probably all of your listeners know about MRSA, right? Methicillin-resistant Staph aureus in the hospitals. And, it's something that, there's a lot of talk about right now. And I think something that, there's going to be, it's expected, deaths will really begin to increase over the next 20 to 30 years due to antibiotic resistant infection. So you'll just go into the hospital, maybe you're going in for something kind of innocuous, maybe you have a small surgery, and you get an infection, and that infection just is with a bacterium that just doesn't respond to any of the antibiotics that we currently have. And the other thing that we're really starting to see is these strains that are actually circulating outside that clinic now, right? So you don't have to go into the hospital now to actually to have these strains infect you. So I think it's something that we're really, there's anticipations. The numbers are, I think the numbers are kind of hard to say, but a lot of folks are thinking there's going to be millions of deaths by antibiotic resistant microbes, and could be tens of millions by 2050. So it's something that, that sort of the alarm bells are starting to go off, and the alarm bells are really starting to go off because we're not really developing a lot of new antibiotics. At least we're not developing a lot of antibiotics that move into the clinic, for sure.
Okay. And so the premise of your research is that bacteria are social animals. What do you mean?
That's right.
What do you mean by that?
Well, so I think it's kind of interesting. I think for a long time, biochemists and folks that were really, the first trying to discover that DNA was the genetic material that helped make up the organism, right? That work was done in bacteria. And at those times, people thought, bacteria were these small entities. We currently, all of us have about 10 to the 12th bacteria per mil in and on our body. I mean, per mil, 10 to the 12th bacteria in and on our bodies. And a lot of people, in the 60s and 70s really thought of these microbes as individual entities. And then about the late 70s into the 1980s, what we learned was that these organisms were actually really social creatures. They didn't act independently. And in fact, we've now learned that they have very sophisticated communication systems where they can actually convey information to each other. And some of the information they convey to each other might be how many bacteria are around me that are like me. They can count their cell numbers. They know what their population numbers look like. They're able to really quickly assess who else is in the community and is this a friend or a foe, right? And then they have very specific behaviors. One thing bacteria can do that obviously that larger animals can't do is they can respond almost instantaneously to new environments, right? So you could actually change their environment, and when that environment changes, they will immediately, within minutes and oftentimes within seconds, begin to adapt very quickly to that new environment. And this ability of them to actually communicate with one another and know who's around them sort of allows them to do this with a little bit more unity than we used to think that they would do this. They didn't do this as individuals. They do this as groups.
Interesting. Okay, so let's just go back in a therapeutic sense.
Mm-hmm.
Why is that important?
So in a therapeutic sense, I mean, it's really important. For example, we work on this bacterium in my academic lab called Pseudomonas aeruginosa. This is a bacterium that produces extracellular molecules that allow it to count its cell numbers. So it, like I said previously, can actually can count how many Pseudomonas are around me. And the reason why this is really important is because most of the toxins that it makes that are actually toxic to humans, it only makes when there's a lot of bacteria around. So if you get a single Pseudomonas into a human, it doesn't make toxins. It sort of sits there and it grows. And the idea is it continues to grow until it gets to a certain cell number. And at that cell number, all the members of the community at once start to make the toxin. And no one really knows, there hasn't been really good experiments to show exactly why this benefits the bacterium, but I think it's pretty easy. Just common sense would tell you that what the bug is doing is it's getting in, and if there's only one of you and you start making toxins and causing trouble in a human, the immune system will find you and kill you, right? There's only one of you. But if you wait till there's 10 million of you, right, and all of you at once begin to make toxins, that's a much, much bigger job for the immune system to actually clear. So the idea is that the benefit of this is that it's to actually overwhelm the immune system and do it all at once. And being able to coordinate toxin production and behavior is the way to do that.
When I first heard you talking about that, what went through my mind, and we were talking about age and that sort of thing before we started here, was the game Risk.
Mm-hmm.
When I used to play Risk, I would sit in Kamchatka, for turns and turns and turns and just collect armies and build them all up there. And then boom, I'm, attacking the world.
Mm-hmm.
Kind of like a bacteria.
Yeah, I mean, it's very, conceptually, it's very simple. And I think, for all your listeners, it makes a lot of sense, right? To actually do this, build up enough resources and enough numbers that you can survive an onslaught, right, from the immune system. And that's what a lot of folks think these microbes are doing. Because, in the early 80s and 90s, when, in the 90s, when I started working on this, most people thought it was really rare. Most bacteria didn't communicate with each other. Most of them didn't really talk to each other. Most weren't social. Now we know that virtually all of them are social. They all are assessing who's around them and counting their numbers and really sort of changing their behavior based on the company, the microbial company that's around them.
So what was the scientific process upon which that conclusion was based?
So how did they discover that? Is that sort of, yeah, so it was actually really interesting. It was actually discovered in a bacterium many years ago, a set of bacteria that are bioluminescent. So there actually are marine microbes. And these marine microbes, what people knew is that if you isolated them and you brought them into the lab and you grew them in a test tube, they would grow really nicely. And then all of a sudden, when they reached a certain bacterial number per unit, so density, they would start to make light and they would glow. You could take them in a dark room and they would glow like a light bulb. And some of these bacteria associated with higher animals and lived in their light organs, and the animals used them for an array of functions. And then it was really that set of bacteria that we discovered were the first ones that were able to count their numbers and really understand and so what they were doing was waiting. And then when the density became high enough, everyone started making light at once. And then there was enough light there that it was of function, right? If a single bacteria, 10 to the minus 15th liters, if one bacterium makes light, there's not enough light there to be of use to anything biologically. But if you have a billion of them that do this, then that sort of then that's enough light that something can actually be used for. And so that really started the field. There was a couple of papers before that where people were alluding to this also. And then people started working on this in human pathogens and showed that the same system that the Vibrio were using were actually being used by human pathogens to actually control toxin production and virulence factors.
So this sociability and environment can cause bacteria to change their behavior.
Yep.
Yes, I think you've discovered that there's a specific environmental characteristic that causes an alteration in behavior. Can you tell us about that?
Yeah, I mean, I think, this idea actually started a long time ago, and we were really, I used to be a test tube microbiologist. I took pathogens out of humans, I grew them in test tubes, and I did studies on them, right? This is kind of how we did science, right? And we really quickly began to understand that that a lot of what we were doing, maybe in the test tubes, wasn't really telling us about what was happening in humans. And the reason for this was just simply that a test tube is not a human, right? It's very different, right? There's different nutrients, right? Just everything about a test tube is very different. And I really started thinking in my academic lab, was like, are we being fooled by this, right? And are we going down these roads of studying? And so what we did, to your question, is we started out saying, well, what are the environmental characteristics, right, that make a human a human? And really we're looking at this from the bacterial perspective, right? What do bacteria sense when they're in humans? And then the question then became very quickly is, could we reproduce that in a test tube, right? And trick the bacteria into thinking they're in a human when they're in a test tube. And we started out doing this, and we found a number of environmental parameters that are really key to this, but a bunch of them are things such as how much oxygen is available, what's available to eat, right? Because bacteria act very, very differently depending on what you feed them, right? And in the lab, what we were doing was just feeding them something we bought from a big pharmaceutical company that said, "Oh, bacteria grow in this," right? And these media have been used for well over 100 years. And then, the very simple question was, could we change that to actually make them more human-like? And that's the real goal that we've been after.
And so, do you think that you could someday regulate that RNA to regulate the level of oxygen, maybe?
Yeah, so I mean, one of the things that we've been, so the first thing the big trick, right, and the big challenge that we actually had was trying to understand what's happening in a human. This is very hard. Like I always tell my undergrads, if you're studying wolves, right, and I said you need to study wolves and you need to really understand wolf behavior, and you want to understand because you want to understand them in a way maybe you want to go in and do some conservation. How would you do those experiments? And this is really easy for us to imagine. They said, well, you would go out to Yellowstone or you would go somewhere and you would put cameras up and you would watch wolves, right? And you'd see what they do when they're in packs, what they do when there's individuals, what they do when they're born, right? How do these behaviors and how does the environment, what they do in the winter and the summer. So, we've been doing this for thousands of years. Aristotle was actually doing this with fish, thousands of years ago. And so we want to do the same thing with bacteria, but it's actually quite hard because bacteria are really small, right? And you can't see them. And plus, we want to do it in humans, right? Where there's a lot of restrictions on what you can actually do and study in humans, right? And so we actually set out. So instead of being able to set up cameras and watch wolves, what we do is we actually go into individuals who have infections, and we actually bacterial infections, and they provide us with a sample from their infection, and we preserve it, and then we sequence the RNA that's in the sample. And then the idea from this is that RNA is the precursor to protein, and protein's what makes us do what we do.
Right.
When you metabolize glucose, it's proteins that metabolize the glucose, right? It's the same thing in microbes. And so what we do is we go out, we take these samples and from individuals, we sequence the RNA, and then from the RNA we say, oh, that bacterium is stressed. It's eating this. It's social right now because it's counting its numbers because we know the genes that are important for that. And we characterize it through this molecular method because we can't use cameras like you would with wolves. And then the idea is from that provided really our first insights into what really are bacteria doing in humans. What is their life like? What's their biology like? And so we were able to discover that, and that sort of served as our gold standard. That now we really understand what's happening in humans, and then we can actually ask, well, now can we make test tube environments that mimic that? And that's been really the idea of whether we can do that. Yeah.
Okay. And so your lab focuses on polymicrobial infections, meaning that there are two or more bacteria occupying the same infection site, making it more resistant to therapy. And you're working, I guess, in three areas. One is gum disease, another cystic fibrosis, and a third is chronic wounds. Let's start with the easy ones. Let's start with chronic wounds. There are some extraordinary numbers in terms of societal impact. Let's talk about that for a minute.
Yeah, I mean, the societal impact of wounds is I think a lot of people don't know this, but it's really astounding the amount of money that just in the US that we spend on healthcare. There's a lot of data out there and people are trying to predict future healthcare costs. And if you look at data from the UK and in the US, I mean, the idea is that by 2050 or so that, maybe between 30 and 50% of the US healthcare budget will be in just basically treating these chronic infections. And the reason being is that, we're generally living to an older population now. But the two big comorbidities for chronic wounds are diabetes and obesity. And these are just skyrocketing right now in places like the UK and the US. And I think most people probably know someone who may be a little bit elderly, maybe has these comorbidities, and they get these infections and these wounds, usually on their feet, right? On their lower extremities. And these are infections that last months, if not years, and are really difficult to treat. And they're often composed, as you said, they're polymicrobial, usually more than one bacterium is there. There could be fungi there also. And there's this consortium of microbes that are living there, and you're just not able to fight them all.
Have you been able to learn anything about those kinds of infections that might imply some new modality of treatment?
Yeah, so I think, as we talked about earlier, the first thing that we really had to understand, which no one really understood, was what were the bacteria in these wounds doing? I mean, were they stressed? What genes were they antibiotic susceptible or not in these infections, and what was their physiology like? And so once we began to know that, we've actually developed some models, some test tube models that you can actually use to study this. And I guess your listeners might not quite understand, why do I always keep saying we want to go back to test tubes? It's because we can do so many experiments in test tubes, and we have such flexibility to really understand something when you're in a test tube compared to maybe a mouse model and certainly in a human.
Right.
And what we've learned is that these microbes actually, they're really interesting. Two of the major players in wounds are Staphylococcus aureus. A lot of these strains are these MRSA strains, and then Pseudomonas aeruginosa. And what we now know is that in these wounds, these microbes actually sense each other very, very specifically. And then when they sense each other, they actually partition themselves and spatially organize themselves very, very precisely. It's kind of like if you just look at a forest, right? There are certain types of trees in one place and others in another. That's what these microbes are doing. They want to be close to each other, but not too close to each other. And they set up these communities that are very well spatially organized. And it's this organization that we've recently shown is what's critical for antimicrobial tolerance. So the reason why they're actually resisting, or one of the reasons why they're resisting when we give antibiotics and put antibiotics on these wounds, we really believe is because of this organization that they've been able to maintain. And so the idea now is, can you make them disorganized? Can you make Pseudomonas and staph not know where each other are so that they can't organize very precisely and spatially organized so that they wouldn't be antibiotic tolerant? And so that's one thing the lab's sort of working on now.
And so what would be, that's interesting. What would be sort of thought of as the way to go about doing that?
Yeah, so you might think, I mean, it's just like a party, right? How do you want certain groups to actually organize within a group, right? And the question is, what cues are they using to do this? So the way that microbes actually know others are around are small diffusible molecules. These are things that are about the size of glucose or maybe a little bit smaller, right? And they're secreting these things out, and the other microbes are detecting them. And they're going, oh, I know that Pseudomonas because when I see these molecules, I know that Pseudomonas, and so then move around. So one way that we're thinking about doing this is to actually, if you just flooded the system with those molecules, there would be no gradients, right? They wouldn't be able to use them, right? And know where to go anymore and how to grow because they don't have directionality. To actually organize yourself spatially, right, in an environment, you need directionality. You need to know where the smoke's coming from, right? You need to know what region, and then to go away from it, right? And so that's the same thing here. But if smoke is everywhere, you don't know where to go. And so that's kind of the idea here is that if we could actually trick them if we knew what molecules they use, which we now are beginning to know, could you trick them, right, into not knowing where they are? Yeah.
Mm-hmm. So help me here for a second, the non-scientist. I read recently about Charles River, which of course has had trouble with monkeys and theft and that sort of thing.
Yeah.
But monkeys, as you've already said, monkeys and mice aren't people.
That's right.
And so they are starting a new venture to try and figure out a way to replace monkeys and mice with something more, something that looks more like a person.
Mm-hmm.
Is it the same kind of thought process from bacteria to?
Yep. So it's exactly the same process that we're thinking about, right? But we're thinking about, instead of thinking about a multicellular organism, we're making test tube models, right? We're tricking the bacteria when it's in a test tube to think it's in a human and act like it's in a human without the human being there. And that's what we've been able to do. And that's what SynthBiome does, is we actually can take a human environment, whether it be the mouth or whether it be a wound or the CF lung, and we're able to make an environment that has in a test tube. And this is just, basically mixing chemicals together. It's kind of like, a witch's brew, right? Of a set of specific molecules that when the microbes get in there, they perceive everything around them and it's human to them. And that's what we've done. A lot of countries, especially, well, certainly in the EU, and I think we're starting to see movement in the US, is away from mouse models and away from a lot of these models, particularly for infection. And the reason being is that mice aren't humans. And a lot of times when you test antimicrobials or virulence compounds in mouse models, the data that you get there is not predictive of what will happen in a human. And so, there's a lot of effort out there. Like I said, Europe really, the EU really led the way, but the US is now following suit, that we need to really think about these models, we need to be able to grow the bacteria in a way that makes them think they're in a human. And the mouse might not be the best way to do this all the time. Sometimes it might. And everyone always says, oh, you're trying to make us not use mouse models. I don't think we will ever not use mouse models, just because if you have a new antibiotic, you have to do toxicity and these kinds of things, right? And that's what we use them for. But I do think that there's a place, a very large place actually, for non-mammalian models that can really be useful for us. And actually, I believe will be more predictive of whether an antibiotic or some antivirulence therapeutic will work in a human.
Right. So at minimum, it potentially compresses the time from thought to market.
Yeah, I think that's true. And test tube experiments are a lot faster. They're a lot more reproducible, and they're a lot cheaper, right? I mean,
Right.
Mice get really expensive, and I can just tell you from our own experience, in the academic lab, when we have a collaborator at Texas Tech. Her name is Kendra Rumbaugh, and basically when we do mouse virulence studies, we do them in our lab and her lab. We use the exact same mice. We brought from the exact same place. They come into Texas Tech. They come into Georgia Tech. We do the experiments with the same strains of bacteria. And oftentimes we can't repeat our results. Like their lab will get something different from us. Not all the time, but sometimes. And it just tells you about the variability in mice, right? These are multicellular creatures, right? That have a large number of microbes that live in and on them. They come into these different facilities. There's different temperatures outside. There's just all of these things, right? And then they respond differently. And this really, when you know there's this much variability in a model that you use, you have to be really careful about how you can take the data from that model and extrapolate later on to humans, I think.
Right. Particularly if it's the environment which is controlling this
Absolutely.
sociability.
Absolutely. Yep.
So then there's periodontitis, gum disease.
Yep.
Tell us about your work there.
Yeah, so that's the fun one, there's not a lot of folks that work on this in the US, but the first bacterium that was ever visualized by a human was from the mouth, right? And Leeuwenhoek actually looked at, he scraped his teeth and used his microscope to look. And, there's, 500 species or so of bacteria in the human mouth. Each of us only has about 150 in our mouths. But these are bacteria that most of them only live in humans, and they have coexisted with one another for thousands and tens of thousands of years. And so these are microbes that are really adept at communicating with each other. They really know who's around them. They have friends, they have foes. They bind to they have proteins that allow me to bind to my friends, and then other ways to get away from my foes, the ones that'll try to kill me. And they form these beautiful communities on our teeth. And we really have used that system to really try to understand, beyond what we already know about communication, what are the other ways that microbes communicate with one another? And periodontal disease is a really big issue, in the US. And I'm not, and a lot of people think about, oral diseases, they think about cavities, right? Caries. You think about your kids having cavities, and, I think, the numbers are 80 to 90% of all humans have at least one cavity in their life, right? So it's a very prevalent disease. But it's really, but that's on your tooth. Periodontal disease is sort of into your gums, between the root of your tooth and your gums. And that's a much more serious type of infection. And the reason is because those microbes have direct access to your bloodstream. I bet probably a lot of your listeners don't know that, most people would tell you virtually every time you brush your teeth in the morning, you introduce bacteria into your blood. And for 20, 30 minutes, maybe an hour, you have bacteria floating around, live bacteria floating around in your blood, which is not a good thing, right? And these are microbes from your mouth. And those microbes have been associated with a lot of different diseases, and a lot of these are still associations, so we have to be careful to know whether they're causal or not. But you're starting to see certainly things, heart diseases for sure, endocarditis. You're definitely, and more recently, I think that you're starting to see some association with Alzheimer's, with a very specific bacterium. And the reason people think this is because a protein, that a bacterium that most of us have in our mouth, ends up in brain lesions, and they found it there. And so again, is it causal or not? We don't know the answer to that yet, but there's a lot of folks that are really starting to gear up. And I think in the next 20 years or so, you're going to see a lot of companies begin to pop up that are focusing on the oral microbiome and the role that it plays in some of these long-term chronic diseases that we have. And it's mainly this idea that these microbes, up in your gum tooth, between your up in the gums by the roots of your teeth, they have direct access to the bloodstream. So when they get in the bloodstream, they can disseminate quite quickly. And so, so in our lab, we're really interested in how this community exists, and then what role might it play in some of these other diseases.
And then finally, there's cystic fibrosis, which is probably where you've had the greatest impact. CF, just so everyone understands, is a genetic disease that makes the patient especially susceptible to chronic infection. How did you think about and later produce a growth medium for the bacteria that closely resembles the human environment in CF?
Yeah, so CF, it's an amazing story, I think, obviously, and it's been a leader in us as humans trying to understand, genetic diseases and ways to actually treat genetic diseases. And it's about 50,000 folks in the U.S. that have this disease, we have, and like you said, they're very susceptible to infections in the lungs. They have a lot of other issues, particularly digestive issues and a lot of other issues. But we focus mainly on the lung infections. It was always very curious that very early on in life they would become colonized in the lungs by very particular bacteria, bacterial community. And one of the most famous and prevalent ones is a bug called Pseudomonas aeruginosa, which I've already told you about.
Right.
It's a really devastating bacterium. I mean, I don't know, but if the listeners can imagine, your lungs are mostly sterile. There's a few bugs down there, but the numbers are actually quite low. And so in CF, for a long time, what was happening was these folks were walking around with billions of bacteria in their lungs. Not millions, but billions.
Wow.
And these are bacteria that are really strong pathogens, Staphylococcus aureus, Pseudomonas aeruginosa. And they would maintain these infections throughout their life. The same strain would sort of stay in there, and it would just evolve with them over tens of years. And a lot of folks were really trying to understand, well, why is this bacterium able to stay in a human infection for 30 years? What is it about that organism? And we were sort of, I think the field was kind of moving forward, but it was a little stagnant. And I think the real reason is because we were taking these strains out of the CF lung and then growing them in the lab in some condition that they've never seen before. And so they acted very differently, and they were fooling us in some ways, particularly my lab. I felt like we were fooled a lot. And so what we did is we actually went in and we said, well, we're going to actually go in and we're going to make a test tube media, a test tube CF human media. And the way that we did this was two ways. Is we actually took samples, so individuals CF for a long time, and it's less now because there are some better therapeutics, but they cough up snot, basically. They call it sputum, but it's snot, right? I mean, like we do when we have viral infections. And we just took this and we just asked the question, what's in it? And we just did chemistry and said, what was in it? We made it in the lab, something that kind of looked like it. And then we put the bacteria in there, and I think they started acting, at least we thought, more like a human. And then we sort of iteratively went through and made changes over the last 10 years. And this was really driven by these experiments that we did that I was telling you about, where we took sputum out of the lung and sequenced the RNA and said, what RNAs are you making in a human? And then what we did is in the lab, our media that we were making, we would go through and we would say, what RNAs are you making? And then we just compared them. And then we said, oh, in the human, these 500 RNAs are different. Well, can we change this medium, our lab that we're working with in the lab, so that those 500 are correct? And we came up with a way to do this. And we've iteratively done this, where now we've developed a medium that is actually, a little over 90% of the RNA in a human is the same in a test tube. And it's very human-like. And now we're at a point where I think you can really go in and understand, because this bacterium in this new medium displays very different behaviors to antibiotics, and it just very different from what we originally thought with it when we were using just our standard cultures.
The sputum medium is really what has so far been the impetus for SynthBiome, right?
That's right.
You're selling it to other labs. How did that happen?
Well, so basically we were doing this, and my lab was really the only lab doing this. And to be honest, we could not get funding to do this from the NIH, the National Institutes of Health. That's the main funder of our labs, But the Cystic Fibrosis Foundation, funds research, and they funded it, and they funded it for almost 10 years for us to continue to do this. And Tim, you would have to realize, science is interesting. A lot of times when you have something new, I think you get a lot of pushback, and we were getting a lot of pushback, and a lot of people were saying this is unnecessary. Why would you do this? This doesn't make any sense. Just use a mouse, like all of these kinds of things. And we pushed and pushed and pushed. And I think, and with the CF, and we wouldn't have been able to do this without the money from them. But we were able to actually go in, and now I really feel like this medium that we've developed is standard in the field. Most people use it. And because most people understand that if they really are trying to understand what's happening in the CF lung and maybe what therapeutic they're developing, how it will it work in a human or not, they understand that this is not, at least most people agree, this is how you test it before you move on. So I think, in the end, to answer your question, it was just persistence, and once you generate enough data, I think, and I think people ultimately will come around.
Can we talk about that persistence for a minute?
Sure.
What happened in year 5 that caused the CF Foundation? What was it that you said to them or did that caused them to say, yeah, we'll go for year 6?
Yeah, these folks, they're, and it's the CF Foundation, but it's also the grant review panel there. And this is a set of folks that I think really want to cure CF. they want to do this, and they realize that microbiology, I feel like the progress was lagging a little bit behind the progress maybe in the cell biology. And I think there were enough reviewers and people on the panel that said we need to take shots at things that some people, that a lot of people think might not work, right? But and I think they're just a little bit more able to do that, I think, and willing to do that, I think, than NIH. NIH is extremely conservative in its funding. They almost want to know it's all worked or it's going to work, right? When they get it, right? For the most part. Now, there are applications that are not like that, but for the most part, they are. But CF, I think you can be a little bit further out there. You can be a little bit more controversial, and they'll fund you. And if you start doing well and they think you're moving forward and you really care about fixing a problem, I think, there's a lot they'll fund you. So I think there's a lot more that goes in. When you get money from a foundation, it's like more like, I feel like a family, right? And you fight about it, right? And maybe you don't agree on everything, but everyone wants everyone to do well, because we want to cure CF, right? And I think that was really, at least for me, I think what sort of maintained our funding through them.
Some of that interest in CF is kind of led, I think, to your move from Austin to Atlanta. Can you tell us more about why you did that, how it happened, and that sort of thing?
Yeah, I mean, I was very happy in Austin. I'm from a little small town outside of Austin. My wife is from Austin. Her dad was a professor at UT Austin. It's a really good school. The science is really amazing, high-end people. But the problem that we began is once we really decided that we wanted to really study human infections, right? I was sort of at a crossroads in my career. I could have stayed in test tubes and figured out how bacteria talk, and I could have figured all this out, right? And I could have kept doing that. But we sort of hit a crossroads, and I really decided I really want to study human infections and see if we can make a contribution there, and maybe try to help folks some. And at the time, Austin, there wasn't a med school there initially. Then there was a med school. There is one now. It's a great med school. It's quite small, but they don't have the sort of research infrastructure and clinical infrastructure that a lot of folks like maybe a UT Southwestern in Dallas has. And so I started looking, but it was hard to find a place because I was doing a little bit of engineering and chemistry and physics and biology. And then I needed some clinical folks, and that doesn't exist in most places. Most med schools don't have the physics and engineering, and most, non-medical schools don't have the clinical parts, right?
Right.
And so, and it was about that time that a job came available here in Atlanta through the Georgia Research Alliance, and it really fit me perfectly because, I could be based at Georgia Tech with the physics and chemistry and engineering and biology, and the science is so good. But then I could be associated with Emory, which is the medical school that has amazing clinicians, right, and a lot of resources. And it really was the perfect kinds of marriage for me, at least. My wife and I were talking about the other day. I don't know that I probably looked at 10 places before I came here. And there's just no place. I don't know if we'd have gone anywhere else. I just think this was the kind of place that kind of fit the bill for us. And so we moved. Yeah.
What a terrific commercial for the bioengineering program between Georgia Tech and Emory.
Yeah, I mean, those schools, there really is, pretty close association, in town, and being close and being able to interact, I think it's pretty remarkable, and plus, UGA is right down the road, Kennesaw is starting to do well. I mean, you have Morehouse. Yeah, I mean, there's a lot, the CDC, obviously, right? There's, if you want to work on infectious disease, I mean, it's a pretty tough place to be.
And so I picture you, Marvin, as kind of the reluctant founder. I mean, you wanted to be left alone to do science, is my understanding. What made you choose to start a company?
Yeah, it was really, for me, I'd been approached multiple times, to start something. it just was so daunting to think that you're running a lab of, you know, a relatively large lab with a fair amount of resources moving through it, and you're running centers and these kinds of things. And then you're going to start a company, but you're going to have to find space. You're going to have to pay rent every month. You're going to have to hire people, right? And pay benefits. And you know what I mean? Like, it was just so much to think about. And I thought I would never have time to do that. And I don't want to do that stuff, right? I just want to do the science. And, you know, and I'll be honest, there's a guy named Tim Richardson here at Georgia Tech. And Tim is like a strategic advisor for Georgia Tech. I think he goes out and he looks for maybe found folks that maybe there's some entrepreneurship in their future or potentially. And he called me on the phone, and I'd had these conversations before, and I was just like, yeah, okay, this is great, right? Sure, we'll start a company, all this stuff, right? And he goes, but Tim was pretty persistent. He goes, no, Marvin, you got to meet these guys. I'm going to put you in touch with these folks from General Inception. And I had a couple of meetings with Vikram and then Paul Conley, you know, who had started GI. And GI is basically a company that starts companies, and they co-found companies with you. And you start a company, and they take care of all the stuff that I don't want to do. And then I get to do the stuff I want to do. And when it first came out, you know, and we first started talking about it, I was like, this kind of sounds a little too good to be true. And when's this? And it kind of was. I mean, they did everything, you know, they provided angel investment, you know, so we could get going. You know, they're in Palo Alto, but they really, you know, they were talking about the company being out in Palo Alto. And I was like, no, I really wanted it in Atlanta. GRA hired me so I would start companies in Atlanta, you know, in Georgia, and then I would contribute to the economy. So we want to stay here. And they were okay with that. It was a lot of paperwork for them, right? And they got all this done, and we found space. And obviously, it's considerably cheaper to get, you know, space out here than it is in Silicon Valley, right? And but plus, I'm a mile away. So if I need to be there, I can be there, you know, at the company if the guys have issues, you know, over there. And yeah, and so, you know, we co-founded, and it's been great. I mean, it's a lot of work, and you know, you know, we've been raising money, and you know, trying to get our footprint into, you know, into ASTs and these other things. And we made a lot of really, I've made a lot of new friends, I think, in that area, which is a lot of fun, and then just learned a lot. So, but yeah, you're right. I was really reluctant, man. I don't know. It was just not something and plus, you know, all your friends, not all my friends, but I've got a bunch of friends start companies. They're, you know, they've all closed for the most part, right? You know, like, yeah. And because they were doing it on their own, and they just couldn't keep their lab going and that going simultaneously. It's too hard.
Right. Do you have any advice for other reluctant founders?
Yeah, I mean, talk to GI. I think that's somewhere. I mean, General Inception's great, but no, I mean, but I think, you know, even though a lot of the paperwork and those other things are still taken off my plate, the one thing I would say is that it still is a lot of work, and it's very different work also. So, sort of the advice that I think I would give is, you know, make sure you have the time for it, right? And if you want to keep your lab going, you know, at your academic home, I think, you know, make sure that, you know, maybe you're in a good position, right? Initially, funding wise, and these kinds of things. And I think that would be one thing, because doing both can be a little bit of a struggle. And then I think, you know, I would talk to someone. You know, we have folks at Georgia Tech, and you know, that you can have conversations with, right, that really let you understand markets and let you do market evaluation. I did not do that, I think, fully beforehand, and I think it would have been nice if we would have maybe a little bit earlier. We've changed our strategic plan a little bit over time, right? You know, our sort of focus a little bit. It's not that different, but it's still changed a little. So if you can do some sort of market analysis and really understand these kinds of things, I think before you go in, it may help you be really focused, because that's what I've learned, I feel like, is being focused and have a clear goal. Science, talking to an academic and to an investor is a very, they're just completely different worlds, right? And it takes a while, at least it took me a while to learn the vernacular I needed to convince someone that maybe we have a good idea.
But what strikes me, Marvin, and what I hope has come through in this interview, is that you have, beyond just the idea, the intellectual ability that all founders in the life sciences have. You're good with people. You like people.
Yeah, I think that's true. It is. I think it is true. I mean, I think there are other people that are probably more comfortable, but I do think that the one thing that I've learned is, look, you have to be honest about things. I think that's easiest. And then I tell my students, my postdocs all the time, that if people can't see that you like it, then they're not going to like it. If you get in front of people for 45 minutes and give a seminar, and it looks like you're like, oh, I can't believe we did this experiment, like this, no one's going to be excited, right? They have to understand that you like it, right? And if that doesn't come through, then I think you've lost before you even start. Right. And so, at least for me, I think, I'd like it, right? I like the science. It's fun, right? I mean, we get to do a job that, I just can't, it's hard to believe we get paid to do it. Like, honestly, that, the way we get paid, it's fun.
Yeah, great. Well, thank you so much, Marvin, for being our guest today. I really enjoyed the conversation and getting to know you. And I hope that others will look to you and apply some of that optimism about the city of Atlanta that you have.
Yeah, well, thanks a lot, Tim. It was a lot of fun. And so, yeah. All right, great. Well, thanks a lot, Tim.
The next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.